Today we're talking about transcription and translation. Here we have our coding and template DNA strands, and under those we've got our mRNA sequence coding for the amino acids. And so the DNA sequence, the way we've got it arranged now, codes for 4 different amino acids, starting with methionine, then valine, serine, lysine, and then it hits a stop codon. And so translation is the act of taking that template strand of DNA and converting it into mRNA. And then from the mRNA, that codes into the codons. Here we've got our first point mutation. So we're going through transversion at the 4th little section. And it's transversion because we're changing between a purine and a pyrimidine, whereas transition is between either 2 purines or 2 pyrimidines. So in this mutation, the new base changes the mRNA sequence, and now it's going to code for asparagine there instead of that lysine. This is known as a missense mutation. Here we have a mutation from AAG to TAG, which is once again a transversion. But unlike last time, our new mRNA sequence is going to code for a stop codon instead of a different, just like functional codon. And so this is going to cause the protein to be halted right there, and the strand is going to stop, and it's going to stop coding. And this is almost always going to be non-functional, just degrade in the cell. This is called a nonsense mutation. For our third mutation, we change from a C to a T, which makes this a transition because it's going between 2 pyrimidines. And so changing it here actually just changes, it mutates. It does not change the amino acid sequence. They both code for valines, although they do it with different mRNA sequences. And so this way the protein will function normally. It'll just have a slightly different base pair. This is called a silent mutation. For our final mutation, we change from a T to an A, which is again a transversion between purines and pyrimidines. And except this one is actually going to happen at our stop codon, which will change the mRNA code from UAA to AAA, which instead of coding for a stop codon and stopping the whole protein sequence there, it's now going to code for another lysine. And so this is known as a read-through mutation, and it's going to lengthen the protein sequence until it reaches another stop codon. And so these are almost always non-functional and will just degrade in the cell. And after the lysine, we've got just the rest of the sequence, and it's got a serine, and it'll just keep on going.